In the name of Allah, welcome to the second part of genetic lecture. Today we are going to talk about laws of genetics. For thousands of years, humans have observed the inheritance of traits from generation to generation and implement their knowledge to breed and domesticate plants and different animals. But the science behind inheritance was only understood in the mid-19th century, when Gregor Mendel, an Australian monk and botanist, published the result of his hybridization studies of pea plant. And he grew and tested over 28,000 pea plants, and in his findings, he noted the role of factors that influence the expression of traits that now we are calling that factors as genes. In this part of the lecture, we are going to summarize the principles of Mendel. The first principle of Mendel is being called as a law of dominance. In law of dominance, for understanding better, let's have a look in this picture. If you remember, we said that we are receiving different alleles from our parents. Suppose if this brown color is a paternal allele and this blue color is a maternal allele. So you can see that in the genotype of that person, there are two different types of alleles. But if you observe the phenotype, or if you observe the person's eye, you can see that the person's eye color is brown. It means in the law of dominance, or in the first principle of Mendel, he said that for each gene, individual or persons are receiving two different alleles, and from these two different alleles, only one of them is dominant. It means one of them is appearing, or one of them is visible, and the second allele would be recessive, or would be invisible or unnoticeable. So we can say that if two alleles at a locus differ than one, the dominant allele determines the organism's appearance. In here, the brown color is dominant allele. The other, the recessive allele, this one, has no noticeable effect on the organism's appearance. The second law, or the second principle of Mendel, says law of segregation. So, if you remember, we said that inside cells, for each characteristics, we have a pair of chromosomes. So, during gamete formation, during formation of sperms and eggs, we know series of cell division occurs, series of meiotic division occurs. During meiotic division, these chromosomes, pair of chromosomes, are segregating or separating randomly. No one knows which of these chromosomes will go to which of the eggs or sperms. This is the law of segregation. The two alleles for a heritable character separates randomly during gamete formation and end up in different gametes. The third principle of Mendel is law of independent assortment, in which he says each pair of alleles segregate independently of the other pairs of alleles during gamete formation. So we know that in each chromosome there are different alleles for the same genes, and during gamete segregation they are independently segregates. For example, look at this chromosome. Suppose if this blue chromosome is dominant over that pink chromosome, but during gamete formation, like after gamete formation, they are segregating independently. It means we cannot say now in gametes, we cannot identify which one is dominant and which one would be recessive. Genetic diagram and Punnett square are two methods used to predict the possible genotype and phenotype among the offsprings of particular cross. So these are Punnett squares and these are genetic diagrams. For using these structures, first of all, we have to separate the gametes in maternal and paternal chromosomes, and we have to write them separately. Suppose these are maternal chromosomes and these are paternal chromosomes. Then we have to cross each of these gametes in a different, and write it in a different square. Then at the end, we can conclude the phenotype and genotype of possible offsprings or filial generations of these organisms. And again, using a genetic diagram, again, we have to separate them to the gametes, and after crossing, we can find out. But the other thing we have to pay attention is, always we are writing dominant allele with capital letter and recessive allele with small letter. When Mendel finished his studies and experiments, he came up with some principles, we said. The first one was the most important one, and he said that for each gene there are two alleles, and from these two alleles, one would be dominant and the second would be recessive. It means in the phenotype of the person, only one of these alleles would appear. But nowadays, scientists discovered that there are some exceptions. For example, some alleles are neither dominant nor recessive. Look at this picture. If we cross a red flower with a white flower, the offspring would be a pink flower. In this situation, we cannot identify the dominant and recessive alleles. So, in this condition, we are facing an incomplete dominance. Incomplete dominance is a kind of dominance in which the offspring exhibits traits that are the blend or mix of the two parents. Or look at this picture. There is a red car and a white car. Their offspring is representing both colors equally. So in this position, again, we cannot identify the dominant and recessive alleles. This is representing codominance, in which when both of the alleles are expressed in heterozygous. The second exception of Mendel principles is that he said that for each gene there are two traits or two alleles, but we have some traits that are controlled by more than one gene. For example, human height, weight, skin color, blood type. These are kind of traits that are controlled by more than two genes. The expression that we can use for these kind of genes are polygenic traits. We discussed about this before. We said that for male and female there are 23 pairs of chromosomes, and these male and females are getting that pair of chromosome from their father and mother. And also we said each of these pairs are representing their unique characteristics, the unique features and unique qualities. But the thing that we have to discuss it now is the last pair of chromosomes, or the 23rd pair of chromosomes, is being called as sex chromosomes because the last pair of chromosomes are determining our gender or our sex. In males, the last pair of chromosome is X and Y, and in female, the last pair of chromosome is 2 X's. So, because the last pair of chromosome is showing, or we can say representing our gender, we can call them as sex chromosomes. We said that after meiosis, while formation of gametes, these two alleles are separating or segregating to eggs and sperm. So after meiosis, when a male makes or produces sperms, the allele for X and the allele for Y segregates in different sperms. The same occurs in the female as well. So after fertilization, if this sperm containing Y allele fertilized one of these eggs, so the result would be a boy. If this sperm containing X allele fertilized one of these eggs, the result would be a girl. About X and Y chromosomes, we have to say that not only they are determining the gender or the sex, they are determining some other features as well. That's why we can call them as sex-linked chromosomes, because usually these chromosomes are located in the X chromosome or in the X allele. So that's why we can call them or refer them as X-linked characteristics as well. The thing we have to know is father can pass X-linked alleles to their daughters but not to their sons, and mother can pass X-linked alleles to their sons and daughters both. Look at this picture. In this picture, you can see that the mother is carrier of hemophilia due to one of her X chromosomes. So if we predict their possible offsprings, we can see that she passed her carrier X gene to one of her sons and one of her daughters. Daughter is a carrier again because daughter receives one X chromosome from the father, which is covering the hemophilia effect of that chromosome. But son is ill. Son has hemophilia because son is receiving Y chromosome from the father, and Y chromosomes cannot cover the effects of X chromosome over here. So we say that in each person there are 36 chromosomes or 23 pairs of chromosomes. What happens if the number of these chromosomes change? Basically, we can understand that any abnormality in the number of chromosomes can cause health issues. For example, if due to cell division, if accidentally or by mistake cell copies 21st chromosomes 3 times, then it can cause to Down syndrome. A person having Down syndrome has these symptoms: small hair at birth, white spots in the colored part of the eye, congenital heart defects, and short, thick hands. They would have short ears and dental problems.